Hey folks, it's me again, your friendly neighborhood biology teacher, Mr. Lang, and today we are going to start our deep dive into photosynthesis with the light-dependent reactions. So I think it's real important that we take 1 second to look at a chloroplast. Remember the structure from the last video and think about where things are happening and exactly what's happening. So if we were to divide photosynthesis into two parts, the first part would be the light reactions and the second part would be the Calvin cycle. The light part, the light reactions part, that's going to be where we're harvesting solar energy, converting it to chemical energy, and then we send that energy to the Calvin cycle where CO2 is going to be converted into sugar. So if we were to look at the energy that's being used to power that Calvin cycle, it's going to be ATP and NADPH. Those things are what are going to be charged up in the light reactions, and that's one way to think about it. Like you charge them up and they go to the Calvin cycle, and then they get discharged, and those can go back to the light reactions to get charged up again and keep driving that sugar production. So we've got our solar farm over here, and we've got our candy factory, our sugar factory over here. So in general, photosynthesis, I'm just going to turn that off. Photosynthesis, like I said, is composed of the light reactions. And that's where energy production is going to happen. We're going to convert solar energy into chemical energy, which has to happen or we can't be alive. The products are going to be ATP and NADPH. Get used to hearing those letters for those compounds because they come up a lot in biology. So ATP is the energy source that drives all of the reactions in our body, and NADPH is a molecule that is used to transfer energy from the light-dependent reactions to the Calvin cycle. So it's integral to everything that's going on in biology because without it, there is no life. And then we have the Calvin cycle. That's that sugar factory time. So we've got, this is called the light-independent reaction sometimes as well, because it can happen without light. You don't need to have photons hitting chlorophyll to make this happen like you do for the light-dependent reactions. And this is where glucose is going to be made. We're making sugar here. This is the energy that we're going to use to make the ATP in our own bodies. And that's what I just said. And sometimes you'll hear it referred to as the dark reactions, but that's kind of a misnomer that you want to stay away from because it's not just happening in the dark, it's happening all the time, whether it's light or dark. It just doesn't need light. So that's why they decided to change that to the light independent reactions. Real safe if you just call it the Calvin cycle. So here is that chocolate factory analogy. So you've got a solar-powered plant that captures energy from the sun, sends that along to Lake Champlain chocolates, and they are making sugary treats for all of us to enjoy. So if you want to think about photosynthesis in this way, it maybe makes it simpler, or we can stick with the complicated stuff, which is what we're going to dive into now. So there's this thing called the electron transport chain. And basically, what is going on is we've got the thylakoid membrane. Remember, don't pay attention to the mitochondria part right now. There's an electron transport chain there as well. We'll talk about that more later on. But right now, we're focused on the chloroplast because that's where we're going to be doing this conversion from solar energy to glucose. So I've got these discs called thylakoids, if you'll remember that from the last time, and a stack of them is a grana. Well, we're looking at one piece of a thylakoid here. So this is that bottom membrane, say right there, where the little box is. So we've got that bottom membrane. And basically what's going to happen is the electron transport chain, which we're going to get into in great detail in just a minute, is going to, in some way, raise the hydrogen ion concentration on the inside of the thylakoid membrane. So inside that flattened disk, we're going to get a high concentration of hydrogen ions. And like you learned in your other bio classes, if you have a high concentration in here, my cursor keeps disappearing, a high concentration in here and a low concentration out here, where does the stuff in here want to go? Well, it wants to leave if it can, and it can through an enzyme called ATP synthase, which does exactly what you might think it does from its name. It synthesizes ATP. Remember, enzymes often end in ASE and have something to do with what their name is. So in this case, ATP synthase drives the conversion from ADP to ATP when you add energy in the form of flowing hydrogen ions that are going to be flowing through. In order to make that all happen, we have to do some things with electrons that get a little bit complicated, but not too bad. Before we start talking about those things, we probably want to talk a little bit about photosystems and what they do. There's two photosystems. There's photosystem 2 and there's photosystem 1. You might ask me why I'm talking about photosystem 2 first. I don't know why they named photosystem 2 when it's the first place that all of the action gets started. It utilizes chlorophyll A, while photosystem 1 utilizes chlorophyll B. They are just slightly different in the absorption spectra that they are best at. So they both absorb mostly red light, which is why they reflect the green light, and that's why plants look green. But basically what's going to happen is we're going to have a photon of light come in, and it's going to get these chlorophyll molecules, these antenna pigment molecules, very, very excited. And as they get, as that excitement grows, there's going to be an electron transfer from chlorophyll molecules that are in the reaction center of the chlorophyll. And I'll show you another diagram that makes that a little more clear in a second. Maybe it makes it more clear, maybe it's just different. Anyways, so we're going to have an electron that's going to be ejected from a chlorophyll molecule, and it's going to go to a primary electron acceptor, and then it's going to be passed along to a bunch of different things in the photosystem to do different jobs. And we're going to talk about what those jobs are in a minute. Oh, hey, there's the reaction center. Great. So here is another look at the exact same thing that I just had up here. So here I've got the photon coming in, energy is being transferred, antenna pigment molecules are getting excited, and then eventually we get to the point where that energy gets to the reaction center chlorophyll and it ejects an electron to a primary electron acceptor. So same idea here. Photon comes in, we pass from molecule to molecule, getting more and more excited. Eventually that excitement reaches those special reaction center chlorophyll molecules, and an electron is going to be ejected out into a primary electron acceptor. So two different ways to look at the same thing. Feel free to use whichever diagram you like better. So important to remember before we look at the electron transport chain itself, that from light energy, it makes ATP and NADPH, and those are the things that are going to go off to the Calvin cycle. It's also important to remember that PS2 and PS1 absorb light and excite electrons, but that only photosystem 2 splits water. So if someone was to ask you, where's water split, you would say photosystem 2. And because that's where water is split, that's also where oxygen is going to be produced. So if somebody said, hey, where is oxygen produced in this whole process? That's it. That's where the oxygen is produced. So I know this diagram looks complicated, but it's not super tough to understand once you've spent a little time with it. So if we look here, before, remember I made a big deal about saying photosystem 2 is really the first step? Well, here's photosystem 2, which is that first step. Light's going to come in, water's going to get split. Here's my thylakoid, just so that we're clear. Light's going to come in, water's going to get split. We're going to build up a hydrogen ion concentration in here, and those hydrogen ions want to leave. They want to diffuse out, and they can through ATP synthase. That's how we make the ATP. It's that simple. Sorry, it's not really simple, but it's that easy. We split the water, we get lots of hydrogen ions. We have some hydrogen ions that get pumped through because of the transfer of the electrons along the electron transport chain. Really high concentration of hydrogen ions in here. They leave. As they leave, they power this ATP synthase molecule to create ATP. Now, a little bit tougher to understand, and we're going to go into some more detail here around how NADPH is produced. So this is photosystem 1. Remember back a couple of slides ago, we had that primary electron acceptor. It kind of even looked like this. So light hits it, excites some molecules. It's gonna hit that reaction site chlorophyll, and that's going to eject an electron, and it's going to go to the primary electron acceptor, which is this plastic quinine molecule, the PQ, which is going to carry that along to cytochrome complex, which is then going to continue to move the electron along the electron transport chain. And we're going to go to ferredoxin, and we're going to go to NADP plus reductase. And that's really where our bread gets buttered in this case. So we're just sort of passing from electron carrier to electron carrier over the course of the electron transport chain. And at each place, we're going to do some things. Here, we're going to use some of the energy to pump hydrogen ions into the thylakoid space so that we can have that ATP synthase down here pumping out ATP. Then the electron's going to come over here, and it's going to get hit by another photon. This is photosystem 1. And the same thing's going to happen. It's going to excite those molecules, and one of the chlorophyll molecules is going to eject an electron to ferredoxin. And that's going to allow ferredoxin to carry the electron over to NADP plus reductase, which is going to take a molecule of NADP plus, and it's going to add 2 electrons, which is going to make it slightly negative, which is going to make it attractive to a hydrogen ion, and all of a sudden we don't have NADP plus, we have NADPH, and that's what's going to go off to the Calvin cycle. We can look at this together. We can answer all sorts of questions about it if that's tough to understand. But one of the things that I like to do is I'd like to look at it in a slightly more simple way. So here's the deal. If instead of looking at this, we look at it happening in this way, we've got photosystem 1, photosystem 2. My computer's doing crazy things. Photosystem 2 and photosystem 1. So a photon of light comes in and starts the reaction, right? That's going to elevate an electron. And we're going to use that to power this mill that makes ATP. That's photosystem 1. This would be ATP synthase. Then that electron is going to continue along to photosystem 1. Another photon is going to come in, and it's going to excite chlorophyll at a slightly different wavelength. And it's going to elevate the electron to an even higher level. And then if there's NADP plus available, NADP reductase is going to convert that NADP plus into NADPH. Sometimes, however, and that's what we want to have happening most of the time, because we want to have both of these things moving off to the Calvin cycle to make sugars for us. Now, that doesn't always happen, though. Sometimes we don't have any NADP plus available. And if that's the case, this electron isn't going to be carried off. It's actually just going to go back into the mix of photosystem 2. And a photon is going to come in, and it's going to excite all the chlorophyll molecules, and eventually an electron is going to get pushed up, and it's just going to keep making ATP. So there's 2 types of phosphorylation going on here for ATP. There's cyclic phosphorylation, and there's non-cyclic. So this would be an example of cyclic, where we don't have NADP plus at the end of the chain. So really, in that case, if it's non-cyclic, we're only going to make ATP. We're not really going to go through photosystem 1, and it's just going to keep rolling, making ATP. The goal is to have NADP plus there and be able to make both products to drive the Calvin cycle. So from there, we're going to take those 2 products, we're going to go to the Calvin cycle, and we are going to create some sugar. and that's going to be the next video, and you're going to love it. So come to class with questions, ask me things, and we'll move on from there. Take a few minutes, think about what we saw, write down things, and rewatch if you'd like. Hope this is helpful.